Renanthera is a genus of large scrambling monopodial epiphytic, lithophytic, and terrestrial species found in India, China, Vietnam, New Guinea, Malaysia, Indonesia, the Philippines and the Solomon Islands. Approximately 20 species of this genus produce a branched inflorescence containing numerous flowers ranging in color from yellow and orange to red, and their flowers possess large lateral sepals. Renanthera imschootiana is the only species in the genus listed in Appendix I of the regulations formulated by the Committee for International Trade in Endangered Species Wild Fauna and Flora. This listing is a result of overcollection for use as ornamental plants or as breeding parents and loss of suitable habitats caused exclusively by human activity in response to the trade of this orchid. Renanthera imschootiana is an extremely rare and endangered tropical epiphytic orchid, which has considerable horticultural value, particularly for its bright colors and long-lasting flowers. Even though it has long-lasting flowers, the percentage of natural insect pollination in Renanthera imschootiana is considerably low. In some cases, almost all the flowers of an inflorescence branch fail to pollinate, which results in zero seed pod formation. The importance of successful pollination in this endangered orchid species cannot be overemphasized. Seed pods are of utmost importance for the efforts required to conserve this species. And, therefore, it becomes very important to understand how to do artificial pollination in this species, and, here in this video, we are going to learn the same. But before we can proceed to learn the artificial pollination technique in this species, let us first get acquainted with its floral parts. Kindly note that orchids have 3 sepals and 3 petals. The perianth segments are showy and colorful because orchids are typically pollinated by insects. One of the petals, called the lip or labellum, is very different from the other 2, and plays an important role in pollination. It often serves as a landing platform for insects. The number of stamens is reduced to 1, and the androecium is adnate to the stigma and style, forming a compound structure called the column or gynostemium. A beak-like structure called the rostellum separates the anther from the functional stigma. A cap-like structure called the anther cap sits on the anther portion. The anther cap is readily detached and is a nice adaptation to prevent self-pollination. This species have compound pollen grain which is hidden by an anther cap, which can be easily removed by a soft tap with a toothpick. In nature, when the anther cap is dislodged by a pollinator, the anther is exposed to the insect's body. Two pollen masses, called pollinia, are attached to a sticky pad-like structure called the visidium. The visidium readily adheres to the bodies of insects. When insects visit anther orchid blossom, the pollinia are transferred to the sticky stigmatic surface just below the rostellum. In order to successfully pollinate Renanthera imschootiana, we are required to manually transfer compound pollen grain into the ventrally oriented gelatinous stigmatic surface. While transferring the pollen grain, it is important to carefully push it into gelatinous matrix of stigma. It is reported that cross-pollination yield more vigorous seedlings than the self-pollination. It is helpful to note down the date of artificial pollination if the seed pods are required for micropropagation. This is because a specific time period known as days after pollination, DAP, is required for optimum growth of seeds. On an average, the percentage of successful seed pod formation after an artificial pollination in this species is between 60 to 80%.